The basic operation process of sample preparation system. 1. Before starting up, check the cleaning solution, shutdown maintenance solution, washing buffer solution, homolyzing, and the reagents inside the reagent tray to ensure all of the sufficient volume. Dispose of waste liquid. 2. Power on. Turn on the power switch. Double-click the RC sampler software on the computer screen to enter the operating interface. 3. Initialization. Click the Initialization button and the device will automatically perform the initialization operation. During the process, no operations are allowed. It takes about 10 minutes until the initialization button to turn blue again, indicating the end of initialization. Open the reagent tree and check if the reagent volume is sufficient. If reagents are sufficient, click Pause and Reagents. Then click the reagent tree to read the reagents, and check again to make sure the reagents are sufficient. Then click Restore and the device will restore to running status. 4. Load the flow tubes and the flow tube drag. Number the flow tubes according to the sample quantity and place them in the flow tube rack. Place the flow tubes into the rack in order from 1 to 12, and the rack indicator light turns green to indicate that the reading is complete. Once again, make sure the number of flow tubes match the sample quantity. 5. End task. Make sure the device is in running or standby mode. Click on Task, New, select the sample type, CD or cytokine. Choose the rack type, capless test tube, capped puncture tube, or EP tube. Select the sample position, rank number, and sample number. Edit sample quantity, hole punching frequency. Select the current operation project. Multiple combinations of the same type of project can be selected, but ensure sufficient reagent volume. Click Save after completing the settings, and the experimental task will be added to the task interface. When ending a new task, make sure the device is running, reagent volume is sufficient, and activate the sample rack and the flow cytometry rack in order to the number which is set before. When ending tasks again, click on the new task interface. Only projects of the current task type can be added at a time. Repeat the process as mentioned above, and the new task will add it to the task interface. Reminder, when the CD type task is running, the cytokine task doesn't allow to end, vice versa. 6. Sample loading. After the new task is ended, place the sample rack into the device loading area. A prompt box will pop up on the testing interface. Click Confirm to start the experiment. Up to 4 racks can be placed at a time. If there are more than 4 racks, they need to be manually loaded again. 7. Experiment starts. The estimated time of completion can be viewed on the task interface. It is not recommended to perform any other operations on the software during experiment. 8. Sample preparation completed. The flow tube rack area turns green and blinks, indicating that samples in the rack have been processed and can be taken out for subsequent operations. 9. Shutdown. After the whole experiment is completed, click Stop and then click Shutdown. Friendly reminder: if reagents will be used the next day, make sure to cap the reagent bottles and put them back in the reagent compartment. It is not necessary to power off. When the reagent are using again, make sure to remove the bottle caps. If reagents will not be used temporarily, take them out and store them in the refrigerator. Close the reagent compartment and power off the device.